वाईफाई ऑफ कर दो, माँ का भी कर दो. Hello friends, good evening and welcome back to the session of biochemistry. We are going to start the nucleotide metabolism today. Can you just give me a feedback? Are you getting the audio clearly? Are you getting the video clearly? Can you see my iPad screen? I hope there is no backlog. There is no sync problem, I hope. Let me know if you're getting the feeds correctly. I'll also start a screen to see if there is any sync problem. Okay, we are live. We are live. Great. So here we go. Here we go. And welcome again to another session of biochemistry. This is probably the second last session of biochemistry that we're going to have. We hope to finish majority of the molecular biology today. Remaining little bit we'll cover tomorrow. And at that time we'll also be giving a quick overview at some of the important techniques that you must know about. If because of any reason the class doesn't happen tomorrow, then we'll finish it the day after tomorrow. All right, so just tell me if you're getting the audio clearly, just tell me if you're getting the video clearly, just let me know if the iPad screen is clearly visible. We are going to start with the nucleotide metabolism today. Okay. All right. So we are starting with the nucleotide metabolism. But before I jump into the nucleotide metabolism, a quick note about the nucleotides that are there in the diet. In minerals, there is only one topic worth covering, and that is iron. So I'll take up some time and quickly tell you the points that you need to know about the iron metabolism, which is commonly asked in the exam. But otherwise, in the last few years, we have not had any question from any other topic except for iron. So we'll try to take out some time and take a look at the iron metabolism and what are the parts that you need to know going into your NEET PG exam. Okay, so talking about the nucleotide metabolism. Before we jump into the nucleotide metabolism, one very important point to keep in mind: what happens to the dietary nucleotides? See, most of our food is organic in nature, and it is coming from the living source, whether it is the plant or it is the animal. So they are likely to contain large amount of nucleic acids, whether it is the DNA or it is the RNA, right? But when we talk about metabolism, we talk about the metabolism of the carbohydrates, we talk about metabolism of the lipids, we talk about the metabolism of the proteins. Hardly you might have noticed that we discuss the metabolism of nucleotides or digestion of the nucleic acids and nucleotides in our gastrointestinal system. So what happens to the dietary nucleotides? Are they digested? Are they not digested? If they are digested, what happens? Are they absorbed? Are they not absorbed? If they're absorbed, do they reach the blood? Do they not reach the blood? So what is the fate of dietary nucleotides? So I'll quickly clarify. The dietary nucleotides are digested, means they are broken down into the simpler components. They are broken down into simpler components. After digestion, they are taken up by enterocytes, but once they are taken up by the enterocytes, here they are degraded. Please note the dietary nucleotides do not reach the blood circulation. The dietary nucleotides do not reach the blood circulation. This I had mentioned when we were talking about the nucleotide analogs. I had told you the nucleotides which are consumed are degraded, but if you modify the nucleotides which are known as the nucleotide analogs, they may bypass the metabolism when they can enter the blood. So the normal nucleotides quickly get destroyed by the enterocytes. They are degraded, and what will reach in the circulation is degradation end products. In the circulation we only get the degradation end products. So if you consume lot of the purines, what you're going to get is a lot of uric acid in the blood. The purines will not reach the blood, rather you will get the uric acid reaching into the blood. So that is very, very important. Second point which I want to make is, what is the storage form of nucleotides? See, we talk about the storage form of the carbohydrates. We say in animals it is the glycogen, in plants it is the starch. We talk about the storage form of lipids. We say the triacylglycerol is the storage form of the lipids. We talk about the storage of the amino acids. We say the proteins in the skeletal muscle can be mobilized when we want the amino acids. Is there such a store for the nucleotides from where you can mobilize them when you need? What is the storage form of nucleotides? Can you mobilize it from the DNA? No, you cannot do that. If you take out the nucleotide from the DNA will be destroyed and the cell will lose its repository of information. Then from where do you get the nucleotides? Please note, there is no storage form. So two things: dietary nucleotides do not reach the circulation. Number two, there is no storage form for the nucleotides. Because of these two reasons, it becomes very, very important that continuous and sufficient synthesis of the nucleotides, both the purine and pyrimidine nucleotides, are continuously occurring in the body, and that is why the nucleotide metabolism is very, very, very important. Let's take a look. Let's start first with the purines and then we'll go on to the pyrimidines. So we're starting with the purines and moving on to the pyrimidines. Okay, so in the metabolism, first we'll talk about the synthesis. Once you cover the synthesis, then we'll go on to talk about the degradation. Okay, so let's first talk about the synthesis. Talking about the synthesis of the purines, we have two mechanisms for synthesis. One is the de novo synthetic pathway, and second is the salvage pathway. So we'll take them up one by one. First the de novo synthetic pathway and then the salvage pathway. Please note again that for most of the metabolic pathways you don't exactly need to know all the reactions. A few, maybe one, two, or at most three reactions may be critical to understand the pathway, but what we need to know is the checklist. So let's look at the checklist for the purine de novo synthesis. We are going to talk about the checklist of the purine de novo synthesis. In our checklist, point number one is, where does the synthesis occur? We just mentioned that the nucleotide must be synthesized. Previously we had said the nucleotides are required by all the cells. We mentioned large number of metabolic processes: the energy transduction, the second messenger, the coenzyme function, transport of the carbon, lot of functions which are required by all cells, and therefore the synthesis occurs in all tissues. If you're asked to mark one tissue, in that case remember, maximal synthesis occurs in the liver. Maximum synthesis occurs in the liver. The compartment where the synthesis occurs is obviously the cytoplasm. The compartment where the synthesis occurs is the cytoplasm. The starting substrate is ribose 5 phosphate. The whole of the purine ring is synthesized over the backbone of ribose 5 phosphate, so that as soon as the purine ring is available, it actually becomes the nucleotide. We don't get the free purine, rather what we get is the nucleotide, because the sugar and the phosphate, the ribose is the sugar and the phosphate is already there, over which we are building the ring. But please note, it is only the starting substrate. The donors are multiple. We have the three amino acids: glycine, glutamine, and aspartate. Glycine, glutamine, and aspartate. In addition, respiratory CO2 is also a donor. Respiratory CO2 is also a donor. In addition, we have the different forms of tetrahydrofolate: the formyl tetrahydrofolate and the methynyl tetrahydrofolate. What each of these give? The glycine will give 2 carbon and 1 nitrogen. The glutamine will give 2 nitrogen. The aspartate will give 1 nitrogen. The respiratory CO2 will give 1 carbon. The formyl tetrahydrofolate will give 1 carbon, and the methynyl tetrahydrofolate will also give 1 carbon. Net 5 carbon and 4 nitrogen. Recall, when we are talking about the purine ring, we had said that the ring contains 4 nitrogen. The ring contains 4 nitrogen. Now, just remembering the donors in terms of carbon and nitrogen is not enough, because you're likely to get this question in the form of a diagram where the individual atoms may be labeled, and you may be asked about the specific source for a labeled atom. So what I'll do, I'll quickly draw the diagram and fill in that information. If you recall, we had started the numbering from here. This would be N1, C2, N3, C4, C5, C6, N7, C8, and N9. The donors: N1 comes from aspartate. N1 comes from aspartate. C2 comes from formyl tetrahydrofolate. N3 comes from glutamine. The N7 C4, C5 and N7, they have a common donor, and if you look at the previous list, it will become obvious. The common donor can only be glycine. The C6 comes from respiratory CO2. The C8 comes from methynyl tetrahydrofolate, and N9 also comes from glutamine. So these are the respective donors in the ring: N1 coming from aspartate, C2 from formyl tetrahydrofolate, N3 and N9 from glutamine, C4, C5, N7 from glycine, C6 from respiratory CO2, and C8 from methynyl tetrahydrofolate. C8 coming from the methynyl tetrahydrofolate. So this is the donor aspect. besides the substrate that you should know about. Once the ring is formed, like I said, it will already be attached to the sugar, ribose, and the phosphate. We have the ribose 5 phosphate. So immediately what you get is the nucleotide. We don't get the free purine. The free purine is not available. We immediately get the nucleotide. So that was the substrates and the donors. The product that we get from the purine de novo synthesis is IMP. IMP stands for inosine monophosphate. It stands for inosine monophosphate. The inosine monophosphate can then be very quickly converted to either AMP or GMP. It can be converted to both the AMP and the GMP. It can quickly be converted to both AMP and GMP. What about the rate limiting enzyme? Please note down the name of the rate limiting enzyme very carefully. We'll revisit this enzyme. The name of the rate limiting enzyme is PRPP, glutamyl amidotransferase. PRPP, glutamyl amidotransferase. This is not the first enzyme, rather it is the enzyme 2 of the pathway. It is the enzyme 2 of the pathway. And then we have the targets for regulation. Then we have the targets for regulation. The targets for regulation are enzyme 1. Enzyme 1 is PRPP synthetase, and enzyme 2, as you've already seen, the PRPP glutamyl amidotransferase. They are the targets for regulation. How are they regulated? The AMP, ADP and ATP, these are the end products from AMP. Similarly, the GMP, GDP and GTP, these are the end products from the GMP. All of them exert a feedback inhibition. All of them exert a feedback inhibition over PRPP synthetase. More are the number of phosphates, the stronger is the feedback inhibitory effect. More the number of phosphates, stronger is the feedback inhibitory effect. Enzyme 2 is inhibited by only two of these molecules, and that is AMP and GMP. Enzyme 1 inhibited by AMP, ADP, ATP, GMP, GDP, GTP in a feedback manner, whereas enzyme 2 is inhibited only by AMP and GMP. The exact energetics is not required, but you should remember the whole process consumes a lot of energy. It is considered to be a costly affair. The whole process is considered to be costly. Multiple reasons. It is a long pathway. We require energy to generate the enzymes of the pathway, and at multiple steps high energy phosphate are consumed. So it is a costly affair, and it is a long pathway. Besides this, what do you need to know? We'll take a look at the first two reactions and then proceed from there. What happens is something like this: the ribose 5 phosphate, the ribose 5 phosphate the phosphoribosyl pyrophosphate, commonly known as the PRPP. The ribose 5 phosphate receives 2 phosphate from ATP. So you'll note the ribose becomes the ribosyl. The first phosphate is the phospho. So phosphoribosyl is nothing but other way of speaking about ribose 5 phosphate, and this pyrophosphate is coming from ATP. The pyrophosphate is coming from ATP. The enzyme catalyzing the reaction is PRPP synthetase. Which class does this enzyme belong to? Just send in the answer in the chat. PRPP synthetase belongs to which class of enzyme? Class 1, 2, 3, 4, 5, 6 or 7? To which class does the enzyme PRPP synthetase belong? Okay, so we get the phosphoribosyl pyrophosphate at the end of reaction 1 catalyzed by PRPP synthetase. If you recall, I had said this is the enzyme 1. Now, one very important point to remember is obviously the PRPP is further metabolized, but in addition to that, the PRPP can go to other directions also. We'll later see. It is very important for the salvage pathway. It is very important for salvage pathway. We'll also see that it is utilized in the pyrimidine synthesis, and later on we'll see that is required for NAD or NADP synthesis. So what is happening? The PRPP can go in multiple directions, just like the glucose 6 phosphate, just like the glucose 6 phosphate. The PRPP can also go in a multiple direction, just like the glucose 6 phosphate. PRPP can also go in multiple directions. Okay, so let's go on to reaction number 2. Going on to reaction number 2. So what happens in reaction number 2? The donor glutamine donates the amide nitrogen. You recall what is amide nitrogen? The nitrogen which has been attached to the side chain. The nitrogen which has been attached to the side chain to the carboxylic group, it is called the amide nitrogen. The amide nitrogen is donated, making the glutamine to glutamate. At the same time, the pyrophosphate is lost. This pyrophosphate drives the reaction forward. It drives the reaction forward, and what we get is the phosphoribosyl. I'm sure you can relate. Phosphoribosyl is coming from the top, and pyrophosphate is gone. What we gain there is the amine. So the product which is formed is the phosphoribosylamine. Obviously, it is catalyzed by enzyme number 2, which was the PRPP glutamyl amidotransferase. Please note, synthetase belongs to class 6 of the enzymes. We had said synthetase and carboxylase are two subclasses which are definitely in class 6. Synthases are mostly belonging to class 4, although they can belong to any class, but the synthetase is obviously belonging to class 6. Same way, the enzyme PRPP glutamyl amidotransferase belongs to class 2, the transferase. Enzyme 2, which is the PRPP glutamyl amidotransferase. Transferase, we had said, belongs to the class 2. It is a class 2 enzyme. So whenever you are doing the carbohydrate, whenever you're doing the metabolism, whether it is the carbohydrate, lipid, amino acid, or any other synthetic pathway, when you come across the enzymes, quickly spend a few seconds and try to locate where that enzyme should be placed. To which class does that enzyme belong? Is it class 1, 2, 3, 4, 5, or 6? So the synthetase belongs to class 6, the transferase belongs to class 2. Now one very important thing which I want to highlight, you must have noticed that we have said that enzyme 2 is the rate limiting enzyme. At the same time, you will notice that both the reactions are irreversible reactions. Both the reactions are irreversible reaction. I have drawn the unidirectional arrow. Now generally the irreversible reactions are rate limiting reactions, but even then, among them the more important rate limiting reaction for the pathway is the reaction 2. Why? Simply because the reaction 2 represents what is known as the committed step. It represents what is known as the committed step. Please understand, in a pathway there can be multiple reactions, but we call a reaction as a committed step when it fulfills the two criteria. Number one, it is irreversible, it cannot go backward easily. At the same time, the product should be used only for that particular pathway. If you go to reaction 1, it is irreversible, so one part of the committed step is fulfilled. But the second criteria, that the product should be used only for this pathway, that criteria is not fulfilled. See, the phosphoribosyl pyrophosphate can go to multiple other pathways. So the enzyme PRPP synthetase is the rate limiting enzyme for which pathway? Is it the rate limiting enzyme for the salvage pathway? Is it the rate limiting enzyme for the pyrimidine synthesis? Is it the rate limiting enzyme for the NADNADP synthesis, or is it the rate limiting enzyme for the purine synthesis? We cannot decide because the product is going into the multiple directions. So although it will be a rate limiting enzyme, it cannot be the most important rate limiting enzyme because it is not limited to the purine synthetic pathway. Therefore, when we talk about the most important rate limiting enzyme, please check it against these two criteria. Number one, it should be irreversible. In very rare cases, this particular checklist may be ignored. Very rare cases. For example, when we talk about the ketone body synthesis, there we ignore this irreversible criteria. All right. And the second criteria is the reaction must be a committed step, meaning the product must be used only for that particular pathway. The product must be used only for that particular pathway. If these two conditions are fulfilled, in that case we can say that we are looking at the most important rate limiting reaction, and therefore the enzyme will be the most important rate limiting enzyme. In this case, it is enzyme 2. Okay, so what has happened? Reaction 1, ribose 5-phosphate gets converted to the PRPP. Reaction 2, PRPP gets converted to the phosphoribosyl amine. What next? After a series of reactions, we finally get the inosine monophosphate. Obviously the intervening reactions are not important. If you count from reaction 1, meaning if you count from the ribose 5-phosphate to the IMP, in all there will be 11 reactions. A total of 11 reactions will be there, meaning the remaining 9 reactions you need not know about. The remaining 9 reactions are not very necessary to know about. Remember, the IMP can then quickly be converted into both AMP and GMP. We already said this AMP can then be converted into ADP and ATP. The GMP can be converted to GDP and GTP. All of these products will have a feedback effect on enzyme 1. The AMP and GMP will also have the feedback effect on the enzyme number 2. In addition, In addition, the GMP promotes the synthesis of AMP. AMP promotes the synthesis of GMP. GMP inhibits its own synthesis. AMP inhibits its own synthesis. This is an additional regulation which occurs. The AMP and GMP upregulate the synthesis of the other molecule. So there is cross regulation. The AMP and GMP cause feedback inhibition of their own synthesis from the inosine monophosphate. This is the second level of regulation which is occurring at the level of substrate. This is the feedback regulation and the cross activation which is occurring. So it will occur very quickly. Remember when we said that substrate level regulation occurs fast within seconds to minutes. It is not the hormonal regulation. Similarly, the feedback effect from the ATP, ATP, AMP, GDP, GMP and the GTP will also occur very quickly. Okay. To summarize, although we call it the purine de novo synthesis, the free purine is not formed. The free purine is not formed. The first nucleotide to be synthesized is inosine monophosphate. What is the nitrogenous base in inosine monophosphate? It is hypoxanthine. The nitrogenous base in inosine monophosphate is actually hypoxanthine. First nucleotide is the inosine monophosphate. Because there are two rings, cyclization reaction must occur twice. Because there are two rings, the cyclization reaction must occur twice. So the question sometimes which is asked is, which ring is formed first, the 5-membered and the 6-membered? Out of them, which ring is formed first? First we get the 5-membered ring. Surprisingly, first we get the 5-membered ring and then we get the 6-membered ring. The 6-membered ring is built upon the backbone of the 5-membered ring. Remember, the purine are attached to the sugar. The purine are attached to the sugar by N9. We had said it is attached via N9 and N9 is present in the 5-membered ring. So we already have the sugar. To the sugar, when the purine will be attached, it has to be attached via N9. N9 is a part of the 5-membered ring. So first the 5-membered ring is synthesized, over which the 6-membered ring will be synthesized. So these are some important points to remember. Free purine is not formed. First nucleotide to be formed is the inosine monophosphate. Cyclization reaction occurs twice. The first ring to be synthesized, the 5-membered ring, followed by the 6-membered ring. So these are some of the important points related to the purine de novo synthesis. Last point, in certain tissues the de novo synthesis does not occur efficiently. In the RBC and in the polymorphonuclear cells, no de novo synthesis. Reason being, the enzyme number 2 is totally absent in the RBC and the mature neutrophils, also known as the polymorphonuclear neutrophils. The enzyme 2 is totally absent. Similarly, in the CNS, the de novo synthesis is greatly down regulated, and again the reason is enzyme 2. Enzyme 2 is produced in very small amounts in the CNS. Enzyme 2 is produced in very small amounts in the CNS. Because of these reasons, for CNS, the salvage pathway becomes the most important source for the purine nucleotides. The salvage pathway becomes the most important source for the purine nucleotides. For RBC and PMN, what happens in case of RBC and PMN? They don't have even the salvage pathway, it is not working at any great level, so they borrow the purine nucleotides from liver. Remember, liver was the site where maximal purine synthesis was occurring, but it is not to meet the requirement, rather it is a byproduct. It is a byproduct and it is waste for the liver. Therefore the RBC and PMN can quickly take it from the liver and reuse for themselves. What is the source of ribose 5 phosphate? If you recall our previous discussion on the metabolism of carbohydrates, you'll remember the ribose 5 phosphate is coming from HMP shunt. It is coming from the HMP shunt. So if the HMP shunt is upregulated, we'll have more of ribose 5 phosphate. If HMP shunt is down regulated, we will have less of ribose 5 phosphate, and depending on the availability of substrate, the pathway, the purine de novo synthetic pathway, can either be upregulated or it can be down regulated. This is very important to understand. If any factor upregulates the HMP shunt, we will get more ribose 5 phosphate and the purine de novo synthesis will be upregulated. If any factor downregulates the HMP shunt, less of ribose 5 phosphate will be formed and the de novo synthesis will also be downregulated. Remember that the nucleotides cannot be stored in our body. So if we have more of ribose 5 phosphate, this will result in more of the purine nucleotides, but they cannot be stored. So they will undergo degradation. And when they undergo degradation, they are going to form more and more of uric acid. So please note, excess synthesis of purines results in excess uric acid formation because the extra purine cannot be stored in the body. Excess synthesis of purines result in excess formation of uric acid because the purine nucleotides cannot be stored in the body, and obviously you know that the excess of uric acid is subsequently going to give rise to the clinical condition called gout initially. Later on there can be other complications, for example there can be stones, there can be formation of tophi, etc., which we will discuss later. Okay, so this is the purine de novo synthesis. We'll do a quick recap for the purine de novo synthesis. Starting with the checklist, we said the synthesis occurs in all the tissues. Maximally, it occurs in the liver. Maximal synthesis occurs in the liver. It is a byproduct because here, if you recall, HMP shunt is upregulated. Liver carries out large amount of synthesis for a lot of molecules for which it requires the reducing equivalent, which will come from the HMP shunt. HMP shunt is upregulated, so we get more of ribose 5 phosphate, and that is why the de novo synthetic pathway is immensely upregulated. The compartment where the synthesis occurs is the cytoplasm. The compartment where the synthesis occurs is the cytoplasm. The substrate is the ribose 5 phosphate. In other words, the starting material is the ribose 5 phosphate, but it is not the donor. The donors are the glycine, glutamine, aspartate, respiratory CO2, the formyl tetrahydrofolate, and the methynyl tetrahydrofolate. Individually, they donate 2 carbon and 1 nitrogen for glycine, 2 nitrogen for glutamine, 1 nitrogen for aspartate, 1 carbon for respiratory CO2, and 1 carbon each for the formyl and the methynyl forms of tetrahydrofolate. Diagrammatically speaking, we saw where each of the donors is giving the atoms in the ring. We are talking about the purine ring. End product is the inosine monophosphate. Recall, in inosine monophosphate, the nitrogen base is hypoxanthine. The nitrous base is hypoxanthine. I mentioned this in the end. The adenosine monophosphate can then be quickly converted either to AMP or GMP by a set of two reactions. In both the directions, we have two independent unrelated reactions which can quickly synthesize the AMP and GMP. The most important rate limiting enzyme for the pathway is enzyme 2, the PRPP glutamyl amidotransferase. It is the most important rate limiting enzyme of the pathway. The targets for regulation are enzyme 1 as well as enzyme 2. Enzyme 1, which is the PRPP synthetase, can be inhibited by AMP, ADP, ATP, GMP, GDP, and GTP. Enzyme 2 is inhibited only by AMP and GMP. Enzyme 2 is inhibited only by AMP and GMP. Remember, the pathway, because it is very long and it requires large amount of high energy phosphate, is considered to be costly. Looking at the first two reactions, the ribose 5-phosphate is first converted to the PRPP, the phosphoribosylpyrophosphate, pyrophosphate, by obtaining 2-phosphate from ATP. The enzyme reaction is catalyzed by the PRPP synthetase. The reaction is catalyzed by PRPP synthetase, the first enzyme of the pathway. PRPP is then quickly converted to the phosphoribosylamine. In this case, the amine donor is the glutamine. This is the amido nitrogen. Enzyme 2 is catalyzing the reaction, which is PRPP glutamyl amido transferase. In the process, pyrophosphate is lost, which is a driving force for the reaction, making the reaction irreversible as well. Please note, the PRPP produced at reaction 1 can also be used for multiple other pathways: the salvage pathway, the pyrimidine synthesis, and the NAD/NADP synthesis. The salvage pathway and pyrimidine synthesis will shortly see. Subsequently, after a series of 9 reactions, the phosphoribosylamine is quickly converted to the inosine monophosphate. Inosine monophosphate can be converted either to AMP or it can be converted to GMP. The AMP exerts feedback inhibition on its own synthesis, just like GMP exerts feedback inhibition on its own synthesis. Also, the AMP and GMP stimulate the production of the corresponding nucleotide from the inosine monophosphate. Points to remember: the free purine is not synthesized because we already have the ribose 5-phosphate. The first nucleotide to be synthesized is the inosine monophosphate, where the nitrogen base is the hypoxanthine. Because two rings are there, cyclization reaction will occur twice. The first ring to be formed is the 5-membered ring, followed by the 6-membered ring. RBC and PMN do not show any de novo synthesis because the most important rate-limiting enzyme PRPP glutamyl amide transferase is absent, whereas CNS shows decreased de novo synthesis because again enzyme 2 is decreased in amount. The RBC and PMN do not synthesize the purine nucleotides even from salvage pathway in any significant amount. They prefer to borrow it from liver, whereas the CNS will carry out the synthesis via the salvage pathway. It is very simple. See, RBC and PMN are mobile. The blood goes to the liver. Liver has a lot of extra purine nucleotides, so we can take up that extra purine nucleotides for use by the RBC and PMN. However, the CNS is sitting in a bony cage. It cannot travel to the liver to borrow the purine nucleotides. Moreover, we have the blood-brain barrier. The blood itself cannot allow the transfer of the purine nucleotides into the brain. So the CNS must synthesize. The only other option left is the salvage pathway. The ribose 5-phosphate, which is the starting material for the pathway, is obtained from HMP shunt. Therefore, any factor which affects the HMP shunt, either causing upregulation or downregulation, is going to affect the concentration of ribose 5-phosphate and therefore the rate of purine de novo synthesis. Please note, if the rate of synthesis is upregulated, we'll get more of purine nucleotides. However, we cannot store the purine nucleotides, therefore they undergo the degradation and the end result is increased level of uric acid. Excess purine production doesn't serve any purpose, rather anticlimatically it results in the production of excess uric acid, which obviously can give rise to gout and other inflammatory ends. That was about the purine de novo synthesis. Let's move on to the salvage pathway. Any questions from the purine de novo synthesis? Please note, the reactions that I've skipped are not vital for your exam. You need not go into the details of those reactions. The first two reactions were important and we have covered them in great detail. The other reactions are not important. You can ignore them. So now we'll be moving on to salvage pathway. If you have any reaction, any questions, any doubts, any part you want me to repeat from de novo synthesis, please feel free to do so. No questions? If there are no questions, then we quickly move on to the salvage pathway. If there are no questions, we'll quickly move on to salvage pathway. Before we understand the salvage pathway, you should know how the degradation of the purine nucleotides actually occurs. Only then we can do the salvage. Salvage is recovery from the waste products. So first we should know the degradation and then we can know the salvage. So degradation is very simple. The nucleotides get converted to the nucleoside. The nucleoside gets converted to the purine, and finally the purine gets converted to uric acid. Now either the conversion from nucleoside to nucleotide or the conversion from purine to nucleotide, both of them will be called the salvage reaction. Conversion from the nucleoside to nucleotide is very simple. We just need a phosphate coming in from any source and we can convert the nucleoside to nucleotide. The conversion of purine to nucleotide in a single step is somewhat tricky. So what we do, we focus on the conversion of purine into the nucleotide in a single step. We are talking about the salvage pathway. The reactions themselves will be called the salvage reactions. The pathway is the salvage pathway and the reaction is the salvage reaction. First point to note in salvage pathway is which purines participate in the salvage pathway. So the purines which participate in the salvage pathway are adenine, hypoxanthine, and guanine. These are the three purines which participate in salvage pathway, and by a single reaction they can be converted to the corresponding nucleotide. Obviously the adenine will get converted into AMP, hypoxanthine will get converted into IMP, guanine will get converted into GMP in a single reaction. How do you convert a purine to a nucleotide in a single reaction? By adding phosphate and the sugar, which can readily be obtained from a PRPP. If you recall, PRPP has the phosphate and the ribose both being present in the form of phosphoribosyl. So the phosphoribosyl group is transferred to adenine and therefore the enzyme is called adenine phosphoribosyl transferase, sometimes written as APRTase. In a single step, adenine quickly converted into the AMP. Exactly the same reaction will occur for hypoxanthine and guanine. Only difference is the enzyme catalyzing both the reactions is the same, therefore it is known as hypoxanthine guanine phosphoribosyl transferase. The full name of the enzyme is hypoxanthine guanine phosphoribosyl transferase. It catalyzes the conversion of both hypoxanthine and guanine to their corresponding nucleotide using the PRPP as the donor of the sugar and the phosphate. PRPP in both the cases will be the donor of the sugar and the phosphate. Okay, what is the benefit of the salvage pathway? First benefit of the salvage pathway is for the CNS. It is a primary source of the purine nucleotide for the CNS. However, please note the salvage pathway can only occur on a background of de novo synthesis. If there is nothing to salvage, how will the salvage pathway work? So even if it occurs at a low level, the de novo synthesis must be going on. Because the de novo synthesis is not going on in the RBC and PMN, it is difficult for salvage pathway to become important in the RBC and the PMN. But in the CNS, some amount of salvage pathway is some amount of de novo synthesis does occur. Therefore, salvage pathway is viable. So remember, the salvage pathway builds up on the de novo synthesis and it is very critical for CNS. It is the primary source of pyrinucleotide for the CNS. Number two, it decreases the amount of uric acid which is being formed. You are taking out the intermediates, so less of the intermediate is available for formation of the uric acid. Therefore what happens is decrease uric acid. Remember, uric acid is sparingly soluble. because it is sparingly soluble. If the concentration increases, it can get precipitated. Unlike the soluble components which do not get precipitated, it can get precipitated, which can cause a large number of problems. Now, in some individuals, the two enzymes may be defective, both the APRTase and the HGPRTase. If only the APRTase is defective, we don't even get to know about it. Why? Because recall, the IMP by a series of two reactions can quickly be converted into AMP. So APRTase does not present to us clinically. There are no signs and symptoms if APRTase is missing, so we don't get to know about it. There might be large number of APRTase deficient individuals, but we don't know because they don't present clinically. However, if HGPRTase is not there, then IMP is not there, and therefore the GMP also cannot be synthesized. This pathway is blocked, the IMP pathway is also blocked, and the de novo synthesis doesn't occur to a significant extent in the CNS. In that case, we are going to end up with a clinical scenario. The absence of HGPRTase presents as the condition known as the Lesch-Nyhan Syndrome. The absence of the enzyme HGPRTase presents as what is known as Lesch-Nyhan Syndrome. The characteristic feature of this disorder is self-mutilation tendency. Self-mutilation tendency. The characteristic biochemical finding is highly increased uric acid levels. Hyperuricemia is almost 2.5 times that of the normal value. The primary cause for increased uric acid is increased PRPP synthesis in the CNS. Increased PRPP synthesis in the CNS. These are the three very important points to remember, which are frequently tested in your exam. The Lesch-Nyhan syndrome is due to absence. Please note, I'm using the term absence. The enzyme is not there at all, totally absence. The absence of the enzyme HGPRTase. Characteristic finding is the self-mutilation tendency, and characteristic biochemical finding is excessive hyperuricemia in the range of 2 to 3 times that of normal value. The primary cause for increased uric acid is the increased PRPP synthesis in the CNS. Let us quickly talk about the Lesch-Nyhan syndrome because it is one of the very important clinical presentations. You are likely to be asked about this again and again in your exam. So, talking about the Lesch-Nyhan syndrome. The Lesch-Nyhan syndrome has clinical features described in the three headings. First, biochemical. We already said in biochemical finding, the primary finding is increased uric acid. The increased uric acid can cause three problematic scenarios. First, gout, that is precipitation of uric acid crystals in the synovial fluid, resulting in inflammation. If untreated, gradually the uric acid crystals will start depositing in the renal calyces, giving rise to renal stones, technically known as nephrolithiasis. The renal stones. And still untreated, gradually the uric acid crystals also start depositing around the joints from the synovial fluid, giving rise to deposits around the joint known as tophi. Tophi is deposits around the joints. So I'll try to show it diagrammatically, see what happens. This is a very schematic diagram. Let's say this is the two surfaces of bone, the articular surface, and this is the synovial membrane. This is the joint space, sorry, this is the joint space, alright, and in which we have the synovial fluid. This joint space where we have the synovial fluid. So initially what happens, the uric acid crystals are getting precipitated in the synovial fluid. They will cause inflammation in the joint space and they will cause gout. But if you don't treat it gradually, the precipitation becomes higher and higher and higher, and these uric acid crystals will increase in amount, and some of it will start seeping outside. Some of it will start seeping outside of the joint space. So if you examine the skin over this space, it appears like a bulbous swelling, like the bulb of the onion. It appears like a bulbous swelling. This bulbous swelling is what is known as the tophi. The bulbous swelling due to deposition of uric acid crystals around the joint, if untreated for long, will cause tophi. Please note, the gout, nephrolithiasis and tophi are not characteristic of Lesch-Nyhan syndrome. They can be seen in any condition causing hyperuricemia which is untreated. Okay, so first is the biochemical category. The second is the set of neurological findings. Second is the set of findings which we call as the neurological findings. Here the first finding is mental retardation, meaning these patients are likely to have poor performance in the school. They will have poor performance in the school, which is the description given in the question. They have mental retardation. In addition to mental retardation, they have varying types of tone disorders. We use the generic term dystonia because in different patients the type of tone disorders may be different. Some patients have athetosis, some have choreathetosis, some have other types of tone disorders. So dystonia is the generic term which is used for the tone disorders in the patients of Lesch-Nyhan syndrome. And the third category is the neuropsychiatric. Third category is neuropsychiatric. First, these patients are very aggressive. The only expression they know is aggression. They love their younger sibling very much. So whenever the younger sibling comes to them, they'll give a tight slap because their affection is being showed by aggression. They know only one expression, and that is aggression. So remember, very, very aggressive. You can have the history frequent suspension from school because the student had a fight with the teacher. So very, very aggressive behavior. And the most characteristic, that is the self mutilation tendency. I'll show you how the typical self mutilation looks like. We have an image showing the self mutilation in a very young child. So the self mutilation tendency is the characteristic finding in the children. Remember, in children, if you find a self-mutilation tendency, the first and the only significant differential which comes to your mind should be the Lesch-Nyhan syndrome. Obviously, in adults, it is different. In adults, the first differential will be a type of schizophrenia, which is called the Van Gogh type of schizophrenia. We will read about it in the psychiatry class, the Van Gogh type of schizophrenia, where the individual can damage himself, sometimes cut off an ear, cut off a part of the finger, something like that occurs in that particular type of schizophrenia. The inheritance pattern is X-linked recessive, so most of the patients are likely to be males. Inheritance pattern is X-linked recessive, most of the patients are likely to be male. In addition, there is what is known as Lesch-Nyhan variant. In the variant, the enzyme is not absent, but it is greatly diminished amount of the enzyme HGPRTase. The resulting condition is known as the Kelly-Sieg-Miller syndrome. The resulting condition is known as the Kelly-Sieg-Miller syndrome. So these are some of the points that you have to remember about the Lesch-Nyhan syndrome. Let me quickly show you the biochemical findings. We said increased uric acid, which can give rise to the gout, nephrolithiasis, tophi. Gout will present as joint pain. The nephrolithiasis, the renal stone will present as the pain in abdomen. Sometimes this pain can be radiating. If the part of the stone dislodges, comes to the ureter, it can be a radiating pain. It can also present with decreased urination or anuria. If it blocks the ureter, then it causes anuria. So these are additional findings which may be given in the question. Abdominal pain, radiating abdominal pain, decreased urination, anuria, these can be some findings. And in tophi, the description will be bulbous deposits around the joint or bulbous swelling of the joints. For mental retardation, typically the description in the question will be poor learning in the school, poor learning outcome in the school, or decreased attention span. These can be some terms which were used. For dystonia, the question, the statement can be abnormal hand movements or history of frequent falls. These terms can be there. For aggressive behavior, you might have a history of frequent fighting among the siblings, frequent quarreling among the siblings, or multiple fights in the school, even with the teacher, leading to frequent suspension. So these can be there. And self-mutilation tendency can be characterized by biting tendency of the hand, the arm, sometimes chewing off a part of the lip. So these can be some of the terms given in the self-mutilation tendency. Typically, these children have a very strong biting tendency. They will keep chewing their fingers, and if you examine a patient at the age of 11 or 12, very frequently you will find that majority of the distal phalanx is completely eroded over the years due to chewing. The children chew off their complete distal phalanx over the duration of 10 to 12 years. So that is the intensity of the self-mutilation tendency. They are biting themselves, the chewing part of the body or biting a part of the body. Sometimes you examine a patient, you'll find a very big bite mark on the hand. It looks like some animal has bit him. When you take the history from the parents will tell you, no, he has bit himself. So the general tendency in self-mutilation is the biting tendency. Biting or the chewing tendency is there, which is directed towards self. It will not affect the surrounding people. Inheritance pattern is X-linked recessive, therefore most of the patients are boys. And there is what is known as the Lesch-Nyhan variant, where the enzyme is not absent, rather it is greatly diminished amount, and the condition is known as Kelly-Sieg-Miller syndrome. In Kelly-Sieg-Miller syndrome, we have the hyperuricemia, but no neurological or the neuropsychiatric presentation. There is no neurological or the neuropsychiatric presentation. The only finding is the hyperuricemia and its sequelae. The hyperuricemia and its sequelae. I'll quickly show you the Lesch-Nyhan syndrome, what it looks like. Let's see if we have the diagram somewhere. Yeah. This is the presentation of the Lesch-Nyhan syndrome. If you will note, nibbled lips and the fingers. You see various parts of the lip which are chewed off. They might even be missing. Just 3 years back in one of the hospitals where I was posted at that time, we had a patient coming in, about 7 years old male child coming in to the emergency in the evening. He had bit off the whole of his lower left lip. A big part from the lower left was missing. The child had bitten it off. So those are the common presentation. In addition, you see the finger is thoroughly chewed, continuously chewing on the finger. And gross bite marks on the hand, you can see the gross bite marks on the hand. The teeth marks on the hand appear like animal bite, but the child has inflicted them on himself. This is what is known as the Lesch-Nyhan syndrome, the characteristic self-mutilation tendency in the children. The question can either be image based, they can give you the image and ask you what is the likely enzyme deficiency, or it can come as a clinical case. Please see, a 10-year-old child with aggressive behavior and poor concentration is brought with presenting complaints of joint pain and reduced urinary output. See, all the factors that we discussed are mentioned in the question: aggressive behavior, poor concentration, joint pain, reduced urinary output. Mother gives history of self-mutilating behavior, stating that he tends to mutilate his finger. So either this clinical case will be there, or the image, the previous image that we saw, this image may be there. And the question may still be the same in both the cases. The question might be, which of the following enzymes is likely to be deficient in this child? So we already mentioned the enzyme which is absent in Lesch-Nyhan syndrome is the HGPRTase. The correct answer in this case will be the enzyme HGPRTase, which is absent in the patients of Lesch-Nyhan syndrome. So that was related to the salvage pathway. Let's quickly go on to the degradation of the purines. We already saw the conversion of nucleotide to nucleoside, so we proceed from the further degradation of the nucleosides. So adenosine quickly converted into hypoxanthine, and this hypoxanthine is then converted into the xanthine. Okay. I'm sorry, we missed one step. The adenosine is first converted to inosine. I'm sorry, we missed a step. The inosine is converted into hypoxanthine, and then we get the xanthine. I'm so sorry for missing one step. Okay. The conversion from adenosine to inosine catalyzed by the enzyme adenosine deaminase. I'm mentioning the name of the enzymes because they're related to certain disorders. That's why I'm mentioning the names. The inosine converted to hypoxanthine by the enzyme purine nucleoside phosphorylase. Purine nucleoside phosphorylase, and the hypoxanthine converted to xanthine by the xanthine oxidase. Also note the guanosine is converted to guanine by the same enzyme, the purine nucleoside phosphorylase, and from guanine it is converted to xanthine by the enzyme guanase. So adenosine to inosine to hypoxanthine to xanthine, and guanosine to guanine to xanthine. In some individuals, the enzyme adenosine deaminase is deficient, giving rise to the severe combined immunodeficiency. There are a lot of causes for the severe combined immunodeficiency, one of them being the adenosine deaminase. It is the second most common cause for the severe combined immunodeficiency. Additionally, in some individuals, the purine nucleoside phosphorylase may be deficient, giving rise to the condition called Nezelov syndrome. Nezelov syndrome is also sometimes considered a subtype of SCID. Not always, but some books will mention it under the subtype of SCID. Now, what happens in SCID and what happens in Nezelov syndrome? In SCID, decreased number of both B and T cells. Whereas in Nezelov syndrome, the number is normal, but there is decreased activity of the B and T cells. The number is normal, but there is decreased activity of both P and T cells. That is the Nezelov syndrome. Coming back to the xanthine is then quickly converted into uric acid, again by the action of xanthine oxidase only. So xanthine oxidase catalyzing two reactions. The first is the conversion of hypoxanthine to xanthine, and second the conversion of xanthine to uric acid. The uric acid is the final end product of the purine degradation in the primate mammals. In the primate mammals, it is the final end product of the purine degradation. However, in a large number of animals, the uric acid conversion is not the final step. Rather, it is acted upon by the enzyme uricase and converted into the water-soluble substance called allantoin. Remember, uric acid is sparingly soluble, whereas allantoin is completely soluble and it is unlikely to get precipitated. Therefore, the better end product is obviously the allantoin and not the uric acid. Uric acid is the end product in primate mammals, but in other animals, we normally get the allantoin. The uricase has been commercialized in the form of rasburicase. For the treatment of gout, it quickly converts the uric acid into the water-soluble form, that is the allantoin. The rasburicase is now clinically available, commercially available. It is used for the treatment of gout. The degradation of the purine nucleotides, adenosine to inosine to hypoxanthine. Enzymes are the adenosine deaminase and the purine nucleoside phosphorylase. The purine nucleoside phosphorylase also acts upon guanosine and converts it to guanine. Hypoxanthine converted to xanthine by the enzyme xanthine oxidase. The guanine converted to xanthine by the enzyme guanase. Xanthine then acted upon by xanthine oxidase to give the uric acid, which is the final degradation product in the primate mammals. It is sparingly soluble. In most other animals, the uric acid is acted upon by the enzyme uricase to give us the completely water-soluble allantoin. Commercially, the uricase is available by the name rasburicase. At this point, I would like to quickly summarize certain important factors which result in increased uric acid or the hyperuricemia. The most common sequelae of hyperuricemia is gout. So we characterize these discussions under the heading of gout. When you talk about gout, gout is divided as per cause into two, as metabolic, whereby there is increased production, and renal, whereby there is decreased excretion. The metabolic gout is further subdivided as per cause. There are conditions where increased enzyme activity will cause the disease. There are conditions where increased enzyme activity will cause the disease, and in this list we have the names. The enzyme PRPP synthetase, and the second name in this list is the xanthine oxidase. If the activity of these two enzymes is increased, they are likely to form more and more of uric acid. In addition, there are conditions where in decreased enzymatic activity can also cause gout. These include the enzyme glucose 6 phosphatase. If you recall, we had discussed this disorder. This is the disorder called the von Wiener's disease. So if glucose 6 phosphatase is not there, the glycogen breakdown will give us a lot of glucose 6 phosphate, which will be directed towards HMP shunt. And now the HMP shunt will become upregulated. Remember what we said, if HMP shunt is upregulated, in that case more and more of purine synthesis, the purine cannot be stored and therefore degraded to give rise to the uric acid. Second condition, deficiency of the enzyme aldolase B, deficiency of the enzyme aldolase B, giving rise to the condition called hereditary fructose intolerance. Here also we find that more and more of purine synthesis occurs, resulting in more formation of the uric acid. Okay, then we come to the secondary conditions which are acquired. The secondary conditions can be subdivided into either there is increased cell turnover, either there is increased cell turnover. For example, we have cancers, we have the myeloproliferative syndromes, condition like psoriasis, or there can be dietary causes, or there can be dietary causes. What are the dietary causes? Excess consumption of fructose. When there is excess consumption of fructose, we had seen fructose metabolism is unregulated, so more and more of glucose will be directed towards the HMP shunt because the fructose will quickly saturate the glycolytic pathway. So increased consumption of fructose, increased consumption of liver rich meat. Remember, liver is one of the primary sites of the purine nucleotide synthesis. We had said the maximum purine synthesis occurs in the liver. So if we consume meat which has more of liver, we are consuming more amount of nucleotide per unit mass, and we had said the nucleotide will get degraded. The uric acid will be released in the blood. And lastly, if there is excess consumption of beer. Why beer? Remember, beer is one of the two undistilled alcohol. So how does it matter if it is undistilled? If it is undistilled, the organisms which synthesize the beer, meaning the anaerobic bacteria and the yeast, are still present in that solution. The suspension that you have contains either living or the dead bacteria and the yeast. And when their body is there, the nucleic acid content is also there. So beer is very rich in nucleic acid content, and when it is metabolized, it will produce a large amount of uric acid. It is going to produce a large amount of uric acid. So these are the primary and secondary causes of the metabolic gout. We move on to the renal gout. Here you should know the metabolic gout, where we said that the disease is due to excess production. These patients are known as overproducers of uric acid, whereas the patients having renal gout are known as under secretors because they are unable to secrete the uric acid which has to be lost in the urine. The metabolic gout makes up barely 15% of the gout cases, whereas the renal gout makes up more than 85% of the episodes of gout. Let's quickly take a look at the causes for the renal gout. By far, the most important cause for renal gout is the chronic kidney disease or the end stage renal disease. The chronic kidney disease or the end stage renal disease. Then there are factors which damage the kidney. For example, the regular chronic consumption of NSAIDs. Most of the NSAIDs would be damaging to the kidney except for the paracetamol. Paracetamol is quite safe in terms of kidney. Then we have specific drugs which prevent the excretion of uric acid, thiazide diuretics. Then there can be situations where the uric acid cannot be excreted due to competition. Any condition which causes lactic acidosis, for example, excess consumption of alcohol. And lastly, the toxicity of lead. Lead inhibits the transporter of uric acid. I'll repeat, If there is any damage to the kidney, CKD, ESRD, the uric acid cannot be secreted here. Please note the uric acid has to be secreted to a great amount in addition to the filtration. Similarly, NSAIDs will cause damage to the papillae and the filtration action will be diminished. Thiazide diuretics will prevent the excretion of uric acid. The lactic acid competes with uric acid for excretion by the same pump. So if there is more lactic acid, less of uric acid is being secreted. And lead also inhibits the pump which secretes the uric acid. The hyperuricemia or gout caused by lead has its own name, and this is known as the Saturnine Gout. This question has been asked multiple times in the exam. Saturnine Gout is caused due to which of the following factors? The answer should be the lead toxicity. The lead toxicity is going to cause the Saturnine Gout. The lead toxicity is going to cause the Saturnine Gout. So these are some of the most well-known causes of gout. I'll not say that this is the exhaustive list, but it covers most of the common causes. It covers most of the common causes for the gout which is likely to land up in your exam. It would be a good idea to revise this before you go for the exam. Very frequently the examiner will try to trap you when they talk about enzyme activity. They will mention which of the following condition enzymes when decreased in amount will cause gout. So you have to be very clear where the increased enzyme activity and where the decreased enzyme activity is causing the gout. This is a very frequently asked question in the exam, and you should be very careful about it. Okay, so that covers our purine metabolism, and from here we go on to the pyrimidine metabolism. Any questions so far? Anything you want to ask before we move on to the pyrimidine metabolism? We are now going on to the pyrimidine metabolism. Any questions so far? Any question, anything you want to ask? So if there are no questions, then we'll go on to the pyrimidine metabolism. If there are no questions, we quickly go on to the pyrimidine metabolism. No questions? Very good. So in pyrimidine metabolism, we have only one pathway that we have discussed, that is the pyrimidine synthesis, and we'll quickly make the checklist. Just like the purine synthesis, the pyrimidine synthesis also occurs in all tissues, and again maximally in liver. Maximally in the liver. The compartment where the synthesis occurs is again the cytoplasm. In this case, the starting material and the substrates include CO2 plus glutamine. These are the starting material or the substrate, and the donor, additional donor, is aspartate. You'd like to see this in the form diagram. I'll draw it quickly in the form of diagram. Recall, we number it from here. This is the N1, C2, N3, C4, C5 and C6. I'll first list them. CO2 will give you one carbon, glutamine will give you one nitrogen, aspartate will give you 3 carbon and 1 nitrogen. So what happens? The C2 comes from respiratory CO2. The N3 comes from glutamine. The N3 comes from glutamine. The remaining structure the remaining structure N1 C4 C5 C6, the remaining structure N1 C4 C5 C6 will come from tetrahydrofolate is not a donor to the ring. The methyl group is getting attached outside the ring. So if the question asks about the donors to the ring, the methyl group, the tetrahydrofolate does not come into the picture. But if it's asked about donor to the pyrimidine molecule, in that case the tetrahydrofolate has to be included. The tetrahydrofolate will be a donor to pyrimidine, but at the same time not a donor to pyrimidine ring. It is not a donor to pyrimidine ring. Okay, so you have to keep this in mind. The methyl tetrahydrofolate is a donor to pyrimidine, but it is not a donor to the pyrimidine ring. Okay, moving on. The end products can be Ctp, UTP, and TMP. If TMP is not in an option, you can mark TTP, but ideally the answer should be TMP. The most important rate limiting enzyme in this case is the enzyme 1. Enzyme 1 is the rate limiting enzyme. The enzyme 1 is carbamoyl phosphate synthetase 2. Enzyme 1 is the carbamoyl phosphate synthetase 2. Which part do you want me to repeat? The target for regulation are both enzyme 1 and enzyme 2. Enzyme 1 we already said is the carbamoyl phosphate synthetase 2. The enzyme 2 is what is known as ATC. I'll write the full form for ATC. ATC stands for aspartate trans carbamylase. It is the counterpart of the OTC. There we had the ornithine transcarbamylase. Here we have the aspartate transcarbamylase. Let's see what happens to these two. What is the regulation? Okay, what is the regulation? Let me finish this regulation and then I'll go back to the ring structure. I'll go back to the ring structure. Let me quickly complete the regulation. The CPS2 is inhibited by UTP. At the same time, it is activated by PRPP. Alright, it is activated by UTP, but at the same time, it is activated by PRPP. ATC is inhibited by CTP, but it is activated by the ATP. It is activated by the ATP. Alright, so you can see there is some level of cross regulation with the purine synthesis also. And very important point to note is the pyrimidine synthetic pathway is regulated by the availability of PRPP. The PRPP right at the beginning will select how much the pyrimidine synthetic pathway should move along, because later on we'll see in one of the reactions the PRPP is directly consumed. So PRPP, depending on its availability, is able to direct the pyrimidine synthetic pathway. This is a very important regulatory point to note. PRPP, depending on its own availability, will direct the pyrimidine synthetic pathway. Just like the purine synthesis, the pyrimidine synthesis is also costly. The exact energetics is not required because it is very lengthy, but a large amount of energy is consumed. What are the additional points you should know here? Unlike the purine synthesis, in pyrimidine synthesis, free pyrimidine is synthesized, which is the orotic acid. The first nucleotide in this case is the nucleotide of orotic acid, orotidylate monophosphate. The first nucleotide is the orotidylate monophosphate. Alright, only one ring, so only one cyclization reaction. Only one cyclization reaction is required, which occurs right in the beginning. About third or fourth reaction is where the cyclization is occurring. These are the additional points to keep in mind. Now I'll quickly go through some of the important reactions. Please note them down. Before I go through the reactions, I'll quickly show the ring structure again. This is the ring structure of the pyrimidine: N1, C2, N3, C4, C5 and the C6. The N1, sorry, C2 donated by the respiratory CO2. The C2 donated by the respiratory CO2. N3 donated by glutamine. The remaining C4, C5, C6 and N1 all are donated by aspartate. All are donated by aspartate. Now there is a special situation, and that is thymine. In thymine, we have to add a methyl group outside the ring. From uracil, conversion to thymine involves addition of the methyl group outside the ring. This methyl group comes from the methyl tetrahydrofolate. So normally when we talk about the pyrimidine ring, when you're talking about the ring, in that case the tetrahydrofolate does not come into the picture, but only for thymine the tetrahydrofolate becomes a donor. So remember, the methyl tetrahydrofolate is donor to pyrimidine, but it is not a donor to pyrimidine ring. So you have to read the question very carefully, whether they are talking about the ring or not, whether they have just written pyrimidine or they have written the pyrimidine ring. If they write the pyrimidine ring, all right, in that case the tetrahydrofolate is not a donor. But if the ring is not written, in that case the tetrahydrofolate will also be a donor. So this is the point which you have to keep in mind, whether the ring is mentioned in the question or not. Okay, let's quickly look at some of the important reactions. The CO2 plus glutamine will combine in the presence of ATP. Here, unlike the urea cycle, the single ATP is required, and the coenzyme N-acetyl glutamine phosphate. Alright, we get the carbamoyl phosphate. The carbamoyl phosphate is acted upon by, I told you, the enzyme aspartate transcarbamylase. The phosphate part is moving out, resulting in formation of the carbamoyl aspartate. the carboyl aspartate. This is ATC. Now the subsequent enzymes are not very important. I'll just keep writing the products. The subsequent enzymes are not important. The carboyl aspartate, here I forgot to mention that there is addition of the aspartate. Obviously there is addition of the aspartate, so you can see all the three substrates or the donors are already added: CO2, the amino group from glutamine, and the aspartate. All of has already come in. The carboyl aspartate gets converted first to dihydroorotate, and then it quickly gets converted into orotic acid. It gets converted into orotic acid. The orotic acid is subsequently converted into OMP by a salvage pathway like reaction. In salvage pathway reaction, what had happened? The PRPP was the donor. I'll write it this side. The PRPP was the donor of the sugar and the phosphate. So something similar is happening here, and the name of the enzyme is therefore also similar: orotidyllate phosphoribosyl transferase. Orotidyllate phosphoribosyl transferase. Alright, and the OMP then gets converted into the UMP. Now the name of this enzyme is very important. If you recall, the name of the enzyme is orotidyllate decarboxylase. Can anyone tell me where we had mentioned this enzyme, the orotidyllate decarboxylase? In the course of our discussion over the last few days, I have mentioned this enzyme, orotidyllate decarboxylase. You have to tell me where I had mentioned the orotidyllate decarboxylase. I'll give a hint. It was discussed in the unit of enzymes only. In the section of enzymes, I had highlighted this enzyme, the orotidyllate decarboxylase. Now this UMP is first converted into UDP, where the pathways can now diverge. There are two possible pathways. One, the UDP first gets converted to DUDP, and the DUDP then gets converted into DUMP. Second possible fate is the UDP gets converted to UTP, the first end product, that is the UTP, and this UTP subsequently gets converted to the second end product, that is CTP. Gets converted to the second end product, that is the CTP. All right. The name of the enzyme catalyzing the conversion from UTP to CTP is the Ctp synthetase. It is the enzyme Ctp synthetase. Now the DUMP, very importantly, gets converted into what we call the thymine or the thymidylate monophosphate. The name of the enzyme being, very good, we had talked about it in the suicide inhibitors. Mamta has given us the answer. I talked about it in suicide inhibitors. Can you tell me what did we mention about the enzyme orotidyllate decarboxylase in the suicide inhibitors? I had mentioned it in suicide inhibitors, but what was the mention? The name of the enzyme is thymidylate synthase. And if you recall, we had mentioned the thymidylate synthase also in the suicide inhibitors. So quickly try to think back, what was the mention for thymidylate synthase and the uridyllate decarboxylase in that question. Okay, for conversion of DUMP to TMP, I already told you that we require the methyl tetrahydrofolate and this methyl tetrahydrofolate in the process is converted into dihydrofolate. Dihydrofolate will first form the tetrahydrofolate, and tetrahydrofolate subsequently converted to the methyl tetrahydrofolate. The reaction of dihydrofolate to tetrahydrofolate is catalyzed by the dihydrofolate reductase. It is catalyzed by the dihydrofolate reductase. So I'll just highlight product 1, product 2, and this is the product 3. These are the end products that we had mentioned. The UTP, CTP and TMP are formed at the end of the pyrimidine synthetic pathway. Now remember, when we are talking about the different types of pyrimidine, we had mentioned that the thymine is present only in the DNA. The thymine is present only in the DNA. Therefore, if I block this reaction, only the synthesis of DNA is going to get affected. Can you tell me the condition where affecting the DNA synthesis only will have lot of beneficial effects? In the meantime, I'll tell you how we can inhibit these two enzymes. If you remember, we had said the thymidylate synthase can be inhibited by the 5-fluorouracil. The dihydrofolate reductase can be inhibited by methotrexate. It can also be inhibited by aminopterin. There are other molecules which can inhibit the dihydrofolate reductase in other organisms, but here the aminopterin methotrexate can be used to inhibit the dihydrofolate reductase. If thymine is not formed, the DNA will not be formed. If DNA is not formed, cell division cannot occur. And can you think of a condition where we don't want cell division to occur? And the answer is cancers. That is why these are used as chemotherapeutic agent. Again, I'll repeat that we are going to have a lot of discussion about chemotherapy in your pharma session. Chemotherapy is a very, you can say, fundamental approach to treat the cancers, and very frequently we are targeting the synthesis of DNA when we are talking about the chemotherapy. The nucleotide analogs are very important in that context. Okay, so very important reaction, conversion of D Ump to Tmp, where enzyme thymidylate synthase and dihydrofolate reductase are involved. They can be targeted to inhibit the formation of TMP and thereby preventing the synthesis of the fresh DNA. Please note that I have not written D in front of TMP, D which stands for deoxyform. I have not written the D TMP in front of TMP. Why? Because as you can see, the TMP is being formed from D Ump. So we only have the deoxy form of thymine. We don't have the ribose form of thymine. We only have the deoxy form of thymine, and that is why the thymine can get incorporated only in the DNA. It has only the deoxyribose form. It doesn't have the ribose sugar. If it doesn't have the ribose sugar, it cannot get incorporated in the ribonucleic acid. That is why we don't find thymine in the RNA. Only exception was tRNA, where the thymine was formed from the incorporated uracil. Uracil was already incorporated and then we modify it to thymine. Okay, so that was the only exception. In tRNA, in the T size EM, we had the thymine which was derived from the modification of the uracil. We had said it is a post-transcriptional modification. It is a modified nucleotide in the tRNA. Okay, so this is about the pyrimidine. Let's quickly do a recap for the pyrimidine. A quick recap for the pyrimidine. We started with the checklist. We said it is synthesized in all tissues. It is synthesized in all tissues, maximally in the liver, just like the purine synthesis, and same way the compartment where synthesis occurs in the cytoplasm. The starting material and the substrates are the respiratory CO2, the glutamine, and aspartate. The respiratory CO2 gives us 1 carbon, the glutamine gives us 1 nitrogen, aspartate gives us 3 carbon and 1 nitrogen, total of 4 carbon and 2 nitrogen. Talking about the structure, we said the C2 comes from the respiratory CO2, the N3 comes from the glutamine, C4, C5, C6 and N1 come from the aspartate. In addition, thymine has a methyl group outside the ring which is donated by the methyl tetrahydrofolate. Coming to point 4, the end products which we just mentioned are the CTP, UTP and TMP. If TMP is not there in the option, you can go with TTP. The most important rate limiting enzyme is the CPS2, the caramel phosphate synthetase type 2. The regulation occurs via the CPS2 and the aspartate transcaramel is slightly complicated. CPS2 is inhibited by UTP but activated by the phosphorybossyl pyrophosphate. Similarly, aspartate transcaramelase is inhibited by CTP but activated by the ATP. So there is some cross regulation between the purines. In addition, there is the feedback inhibition. See, feedback inhibition here, feedback inhibition here. Just like purine, the pathway is costly. We summarize that free purine is synthesized, which is free pyrimidine is synthesized, which is the orotic acid. The first nucleotide is the orotate monophosphate. Only one cyclization reaction is required. Only one cyclization reaction. We had seen conversion of dihydroorotate to orotic acid. Seeing at the reactions, the reaction 1, conversion of CO2 to glutamine, conversion of CO2 plus glutamine to carmyl phosphate, conversion of carmyl phosphate to the carmyl aspartate. These two are the critical reactions that you must know about because they are targeted for regulation. Moreover, enzyme 1 is the most important rate limiting enzyme. Carmyl aspartate converted to dihydroorotate, dihydroorotate converted to orotic acid. This is where the cyclization is occurring. The orotic acid converted to OMP and the OMP is then converted to UMP. This is a secondary target for suicide inhibition by the allopurinol. The OMP is then converted, the UMP is then converted into the UDP. From here, the UDP can go in two directions. Either it can get converted first to UTP and then to CTP, or it can get converted to deoxy UDP and then the deoxy UMP and finally the TMP. Okay, in the conversion of DUMP to TMP, two very important enzymes are involved, the thymidylate synthase and the dihydrofolate reductase, both of which can be targeted by 5-fluorouracil methotrexate as part of chemotherapy. As part of chemotherapy, the thymine synthesis, if it is blocked, will prevent the synthesis of the DNA. It will prevent the synthesis of DNA. Any questions so far? No questions? Great. If there are no questions, then one extra point, bonus point that I want to tell you. Most of the reactions that we have done, we saw that the sugar which is there is the ribose sugar. But to make the DNA, we need the deoxyribose form. We saw the deoxyribose form only in the thymine. So what is the mechanism to generate the deoxy form for the other nucleotides? And that occurs in the following manner. We must have the nucleotide diphosphate. We must have the nucleotide diphosphate. Alright, the nucleotide diphosphate is acted upon by the enzyme nucleotide reductase. Nucleotide diphosphate is acted upon by the nucleoside reductase, resulting in the formation of deoxy nucleotide diphosphate. Rather, I should use the term nucleoside, not the nucleotide, because the two phosphates are mentioned separately. The nucleoside diphosphate is converted to the deoxy nucleoside diphosphate. Now, in this process, the reducing equivalent is provided by a substance called thioredoxin. The reducing equivalent is provided by a substance called thioredoxin. Initially, it is in the reduced form and it provides its reducing equivalent, converts the ribose sugar to the deoxyribose sugar, and gets oxidized. Itself gets oxidized. It is recycled by the enzyme thioredoxin reductase. If you recall, we had mentioned this enzyme at one place. Anyone can tell me where we had mentioned this enzyme, the thioredoxin reductase? Anyone? The recycling of thioredoxin reductase reduces receives the reduced equivalent from NADPH. Okay, so again, for antioxidant action, recall that we had said NADPH is required. For antioxidant action, the NADPH is required. This is how the nucleosides can be converted to the deoxy form. One very important thing to know, we must have the diphosphate form. If you have the monophosphate form, if you have the triphosphate form, the nucleotide reductase will not act. Nucleotide reductase is very specific. For the diphosphate form, it is able to act only on the diphosphate form. The initial reducing equivalent provided by the thioredoxin, ultimately the reducing equivalent is coming from NADPH. Ultimately the reducing equivalent is coming from NADPH. Okay, so this is last point about the pyrimidine as well as purine synthetic part. A little bit about the pyrimidine degradation. We'll not go into the details of pyrimidine degradation simply because the end products are water soluble, so the degradation pathway is not important. It doesn't generally cause any disease, but what you should know is end products of pyrimidine degradation. What are the end products of pyrimidine degradation? You can remember it by the mnemonic A B to C. End product of pyrimidine degradation remembered by the mnemonic A B to C. A stands for ammonia. First B stands for beta alanine. The second B stands for beta amino isobutyrate. Second B stands for the beta amino isobutyrate. And the C stands for carbon dioxide. So A, B2C, ammonia, beta alanine, beta amino isobutyrate, and carbon dioxide, they are the end products of pyrimidine degradation. Please note, all of them are freely water soluble. All of them you can see are freely water soluble. In addition, one extra point to note, the beta alanine and beta amino isobutyrate are beta amino acids. When you're talking about amino acids, we had mentioned the alpha amino acids, but these are beta amino acids. Beta amino acid means the amino group is attached to beta carbon. What do you mean by beta carbon? I'll show the structure. See, normally what happens, this is the carboxylic group. Where the carboxylic group is attached, we call it the alpha carbon, and the amino group is also attached here. Alright, so this will be the alpha amino acid. But when I say beta amino acid, the amino group is attached to a carbon after that. So this is the beta carbon, and the amino group is attached to the beta carbon, and therefore we call it the beta amino acid. Please note, the beta alanine, beta amino acid butyrate are instances of naturally occurring beta amino acid from the degradation of the pyrimidine. From degradation of pyrimidine we get the beta amino acid. In addition, there are some disorders First, we have the inherited disorders of pyrimidine metabolism. Inherited disorders of pyrimidine metabolism. The two types of orotic aciduria, type 1 and type 2, involving the reactions 5 and 6. Involving the reactions 5 and 6, if you recall, the reaction 5 was conversion of orotic acid to OMP, and reaction 6 was conversion of OMP to the UMP. So if those two enzymes are missing, then we get the orotic aciduria type 1 and type 2. In addition, we have what is known as the beta hydroxy butyric aciduria. So the formation of beta amino acid butyrate is blocked at the level of beta hydroxybutyric acid, and this part accumulates in the body and it can be released in the urine, giving rise to the beta hydroxybutyric aciduria. Also recall that orotic aciduria occurs in urea cycle disorders, although there is no defect in the pyrimidine metabolic pathway. Still in the orotic acid, still in the urea cycle disorders, we get the orotic aciduria. Why? What is happening? There is accumulation of the carboyl phosphate in urea cycle disorders. The carboyl phosphate will come out from mitochondria into the cytoplasm. Once they are in the cytoplasm, they have bypassed the regulatory step by PRPP. If you recall, PRPP stimulates enzyme 1, which synthesizes the carboyl phosphate in the cytoplasm. But if we get the carboyl phosphate from the mitochondria, we have bypassed the regulation by PRPP. What is the net effect? PRPP we had seen was required at reaction 5, where the orotic acid is getting converted into OMP. There the PRPP was the donor of the phosphate and the ribose sugar. Now, depending on amount of PRPP which is available to participate in reaction 5, there is upregulation of reaction 1. When you bypass the regulation at reaction 1, what will happen? You will have more of orotic acid and less of the PRPP. So it's like you have made the food for 100 people, you have invited 100 people, but 150 people land up, so 50 people are going to go hungry. So whatever extra carboyl phosphate comes from the mitochondria, it can go right up till orotic acid, but it cannot get converted into the OMP, and that is why we find the orotic aciduria in the urea cycle disorders. So this is the list of inherited disorders of pyrimidine. We also have certain instances of acquired orotic aciduria. Only for orotic aciduria, there are certain instances of acquired orotic aciduria. First is seen in children. In children, we see what is known as the Ray's syndrome. You know, the excess consumption of the acetyl salicylic acid causes damage to the kidney in the children. It is known as the Ray's syndrome. What happens when there is damage to the liver? The damage first occurs inside the mitochondria, because the acetyl salicylic acid is metabolized in the mitochondria. When the mitochondria is damaged, the first appearance is the increase in permeability of the inner mitochondrial membrane. The carboyl phosphate, just like in the urea cycle disorder, will start coming out. It will participate in the pyrimidine synthesis and it will get blocked at the orotic acid level, and orotic aciduria will occur. In adults, we see what is known as allopurinol toxicity. This can happen when you have a very intelligent patient. What happens, as a clinician, you do the upward titration of allopurinol when you are trying to treat the gout. You start with low level, you observe the patient for 2 weeks. When there is no fresh episode of gout but still the uric acid level is high, you increase the drug, observe for 2 weeks. When the levels stabilize, still the uric acid is high, you keep increasing the drug till you reach the maximum dose of maximum dose of 900 milligram per day for a standard meal. 900 milligram per day is the acceptable dose. So what happens on first visit, you ask the patient, okay, you start taking 100 milligram twice and come back after 2 weeks. If there is episode in between, you come in between. So 2 weeks, now you tell him you start taking 200 milligram per day twice, and then you come back to me after 2 weeks. So after 2 weeks again the patient comes, there is no episode of gout in between. You check the levels are still high. So then you shift the patient to 300 milligram twice daily, and you tell him you come back to me after 2 weeks. After 2 weeks again the patient comes, there is no episode of gout in between, but the levels are still high. So you go to the maximum permissible dose. You tell him start taking 300 milligram thrice daily, and that's it. We'll see what we have to do next. You don't call the patient back after 2 weeks, because now you know you cannot increase the dose beyond that. You tell him when there is episode of gout in between, then you come, otherwise you come maybe after 3 months. So the patient goes back home, starts taking the allopurinol thrice daily, and after 2 weeks he suddenly realizes, oh, you have not called him back after 2 weeks. You told him to come back after 3 months. So he thinks, okay, maybe doctor has forgotten to call me. So what he does, I'll do the same thing that the doctor does. What the doctor does, every time I visit him, he increases my dose. So the patient will increase the dose by himself. He'll start taking the allopurinol 300 mg 4 times daily, and that is when he will land up in allopurinol toxicity. And if you recall, what enzyme will be inhibited when there is allopurinol toxicity? The enzyme which will be inhibited is enzyme number 6, orotidylate decarboxylase. Result, accumulation of OMP. Accumulated OMP will result in accumulation of orotic acid, which will start appearing in the urine, resulting in the orotic aciduria. So I'll quickly talk about the end products of pyrimidine degradation disorders. We can remember it by the mnemonic A, B to C: ammonia, beta alanine, beta amino acid butyrate, and the carbon dioxide. The beta alanine and the beta amino acid butyrate are the beta amino acid where the amino group is attached to the beta carbon. The inherited disorders of pyrimidine metabolism include the orotic aciduria type 1 and 2. Affected enzymes are reaction enzyme 5 and enzyme 6, and the beta hydroxybutyric aciduria. We also saw when we are doing the urea cycle disorders that orotic aciduria is seen in a majority of the urea cycle disorders except for two. The orotic aciduria is seen in all the conditions. We can also have two situations where we find the acquired orotic aciduria. These two conditions are, first, in children we have the Ray's syndrome, where there is damage in the mitochondria of the liver and the carbonyl phosphate from the liver comes out into the cytoplasm. In adults we can have the allopurinol toxicity, where allopurinol will inhibit the enzyme orotidylate decarboxylase, giving rise to accumulation of orotic acid. So that concludes our discussion of the purine and the pyrimidine. Any questions so far? Anything you want to ask? Any question you want to ask? Okay, so at this stage we are left with the synthesis of the DNA, RNA and the protein, and a little bit about the regulation. So we are going to take up those topics tomorrow. We'll also talk a little bit about the uh, iron metabolism then, but in the meantime I want you to answer a few questions. First, please note down the name of the stop codons. The names of the stop codons. If you talk about the RNA, then the name is UAG, but in the DNA it is not U, it is T. So on the coding strand on the DNA we'll have the TAG. We'll have the TAG. So this is known as amber. This is known as amber. The UAA, this is known as occur. For the DNA, the corresponding codon will be TAA, and UGA is known as opal. For the corresponding codon on the DNA, it will be TGA. This question has been asked sometimes in the past, about 3-4 times. This question has been asked: which of the following is not a stop codon, which of the following is a stop codon, which of the following is the correct name for the stop codon, UAG, like that. So those questions have come, so you should know these names and to which codon do they correspond. Okay, so these are the name of the stop codons. Moving on, first question: the histone and the purine, which of the following is not a part of the nucleoside? Please send in the answer. Which of the following is not a part of the nucleoside? The pyrimidine can be a part. It is one of the nitrogen bases. Similarly, sugar. See it? It is not seen in the nucleoside. You will see the histone in nucleosome. Very good. Very good. All of you have given the correct answer. The answer is 3. Histone is seen in the nucleosome, not in the nucleoside. Okay. Moving on to next question. The concentration of DNA. Concentration means you want to find out the quantity. The concentration of DNA is measured by which of the following steps? Either you do the infrared examination of the solution, you do the deoxyribose estimation, you measure the absorption at 260 nanometer, or what you do the ultrasound examination of the solution. Which of the following will tell us the concentration of DNA? Basically what you're doing is quantification. We are doing the quantification of DNA, because once you know the concentration, using the standard conversion factor, you can calculate the amount of DNA in the solution. Amount of DNA can be calculated, and the correct answer is, we have already mentioned, absorption at 260 nanometer. Absorption of 260 nanometer is used for quantification of not only DNA, for any nucleic acid, whether it is DNA, whether it is RNA, or it is the nucleotides. All of them can be quantified by measuring absorption at 260 nanometer, because 260 nanometer is the absorption maxima for nucleotides. Very good, very good. The correct answer is 3, measuring the absorption at 260 nanometer. Moving on, question number 3. The two strands of DNA are held together by which of the following bonds? The van der Waals bond, the hydrogen bond, the covalent bond, and the ionic interaction. Please note, the van der Waals bond is non-covalent. The hydrogen bond is also non-covalent. The ionic interaction is also non-covalent. So which of the 4 bonds will hold the two strands of DNA together? If you recall, the two strands of DNA are held together by a type of bond between nitrogenous bases. So you have to tell us which type of bond is present between the nitrogenous bases by which the two strands of DNA are held together. Very good. The correct answer is the hydrogen bond. Very good. The correct answer is the hydrogen bond. This question has also been asked in the form of diagram. If we have that question, I'll show you how it can be asked in the form of the diagram. Now read all the options carefully. Apply the formula that we have determined. Put in the values and quickly check which of the options is correct. You must check all the options before you mark the answer. So the question is, the Chargaff rule states that A plus G is equal to T plus C, A upon G is equal to C upon T, A is equal to U is equal to T is equal to G is equal to C, A plus T is equal to G plus C. Recall that here we have to use the two equations, A which say one which says A is equal to T, and 2 which says G is equal to C. In other words, you have to quickly replace A by 1, G by 2, T by 1, and C by 2. All A, let's replace all A by 1, all T by 1, all G by 2, all C by 2, all G by 2, all C by 2. T and U are analogous, it will be 1, G by 2, and C by 2. So what should be the correct answer? Quickly calculate them. First option says 3 is equal to 3. Second option says 1 upon 2 is equal to 2. Third option says 1 is equal to 2. Fourth option says 2 is equal to 4. So obviously the correct answer should be 1. Only equation 1 is correct as per our rules. All other we can ignore all the others. Okay, moving on to the next question. At physiological pH, the DNA molecules are positively charged, negatively charged, neutral, amphipathic. Now two things here. First, when I say physiological pH, what I mean is the pH of 7.4. Second, a lot of times you'll get the question like this where more than one answer is correct. In that case, you have to choose the best possible answer. A lot of time you'll get the situation where more than one answer is correct. You have to choose the best possible answer. I'll give you the hint. The negatively charged part is correct, the amphipathic charge part is correct. So out of the two, the better answer is obviously the negatively charged, because it depends on the pH. The amphipathic nature does not depend on the pH. Alright, so when we're talking about the pH, out of the two properties, the negatively charged becomes more important. The negative charge is due to the presence of the phosphates. The negative charge is due to the presence of the phosphates. Okay, so the answer is negatively charged. Next question, which form of DNA is predominantly seen? Which form of DNA is predominantly seen? Your options are Z form, the A form, the C form, and the B form. Alright, these are the different conformations which are seen. Obviously, the most common form is the B form. The Z form is the second most common form. So the most common form is the B form. Moving on to next question. See, I told you we will get the diagrams based question. What is the nature of bonds between the two nitrogenous bases on opposite strands of double-stranded DNA? The arrow is marking to the point between the two nitrogenous bases. You can see the dotted line is there between the two nitrogenous bases. The dotted line is there between the two nitrogenous bases, and you have to tell us what is the nature of bond. So this question we had discussed previously. The bond between the two nitrogen bases is obviously the hydrogen bond. So the correct answer is option 3, the hydrogen bond. Okay, moving on to next question. Intron is not found in which DNA? Intron is not found in which DNA? Your options are the nuclear DNA, the mitochondrial DNA, the B form of DNA, and the Z form of DNA. Intron is the part which is not translated, found in between the exons. It has to be removed before we can carry out the translation. And we have discussed the intron is not found in the mitochondrial DNA, or you can say it is not found in the prokaryotes, or you can say it is not found in the bacterial DNA. Okay, so the correct answer here will be the mitochondrial DNA or the prokaryotic or the bacterial DNA. In the human DNA, rather in the nuclear DNA, which particular gene does not have introns? Remember, in the nuclear DNA, only histone genes do not have intron, all others are going to have the intron. All others are going to have the intron. Very good. The correct answer is the mitochondrial DNA. Moving on to next question. The protein rich in basic amino acids which functions in the packaging of DNA in chromosomes is option 1 elastin, option 2 collagen, option 3 histones, option 4 fibrinogen. Remember, DNA is negatively charged, therefore to package the negatively charged DNA we need the positively charged, or you can say the basic protein, and that is the histones. We've already done this. Together they form the nucleosomal structure. 1.75 loops of DNA are wound around the histone to form one nucleosome. We get the histone octamer around which 1.75 loops of DNA are present. Very good. The answer is option 3 histones. Option is answer is option 3, that is histones. Moving on, euchromatin is the region of DNA that is relatively, you have to choose which is the correct description for the euchromatin. Euchromatin is uncondensed, it is condensed, it is overcondensed, it is partially condensed. First you should remember, euchromatin is the part which is undergoing transcription. We had mentioned it is the part which is undergoing the transcription. For transcription to occur, the two strands of DNA must be separated. When you separate the two strands of DNA, the DNA gets uncondensed. Alright, the others are not the correct answer. We had mentioned the euchromatin is uncondensed. The euchromatin is lightly staining, it is uncondensed, it is transcriptionally active. These are the three features of the euchromatin we had mentioned when we are comparing with the heterochromatin. Very good. The correct answer is uncondensed. Correct answer is uncondensed. If you check your notes going back, you will find we have written it is uncondensed. Coming to the next question. The question is based on the mitochondrial DNA. It asks the mitochondrial DNA is inherited in what manner? It is horizontally inherited, it is vertically inherited, it is paternally inherited, and it is maternally inherited. What are the ways? What is the correct answer? We had mentioned the mitochondrial DNA is inherited only from the mother, so the best answer is maternally inherited. Horizontal inheritance example is conjugation in bacteria, transfer of plasmid from HFR positive to HFR negative bacteria. This is the example of horizontal inheritance. Horizontal inheritance means the genetic material has to travel between two organisms of the same generation. Okay, so that is the horizontal inheritance. Here we are talking about the maternally inherited. If maternal inherited is not there in the option, then you can mark the vertically inherited, because maternal inheritance, paternal inheritance both are vertical inheritance. If the maternal inheritance is mentioned, it is obviously the better answer. If it is not there, then the alternate answer can be vertically inherited. Maternal inheritance is a type of vertical inheritance. Okay, moving on. Non-coding RNA are all except, your options are silencing RNA, the microRNA, the transfer RNA, and the messenger RNA. Now this is a tricky question, that's why I placed here. This is what we know as a double negative question. Although the examiners get the instruction, do not create double negative question for the exam. Every time you will find about 10 to 15% of questions which are double negative. Now double negative questions are very simple to answer if you are careful. If you are careful, they are simple to answer. What you have to be careful about, you should be able to correctly identify the negative instances. So first negative instance is the non, second negative instance is all except. If you correctly identify them, all right, then the question becomes coding RNA are, and we have already written coding RNA is only one. The coding RNA is the messenger RNA. Very good. So the correct answer coding RNA or the messenger RNA. So please be careful when you get the double negative question. They are the questions where we most commonly make the mistake. If you are able to identify a double negative question, try to remove the double negative clauses so you can get a more clear question. The clear questions are easy to answer. Otherwise, you are likely to make a mistake if you try to answer it without resolving the two negative portions. Very good. I was happy to see that all of you gave the correct answer. Correct answer is the messenger RNA. Moving on to next question. What is the normal role of microRNA? What is the normal role of microRNA? Your options are regulation of gene expression, the RNA splicing, initiation of translation, the DNA conformational changes. What is the normal role of microRNA? If you go back to your notes, you will find we have mentioned the function of microRNA. 100 messenger RNA can be regulated by a single microRNA. So the correct answer is regulation of gene expression. Correct answer is regulation of gene expression. Very good. Murtaza is regularly giving us a lot of correct answer. I'm so happy all of you are. Your options are the DNAs, the helicase, the small nuclear RNA, the small nuclear RNA. Oh shit. What happened? The screen is gone. Phew. There was a very short technical error. Our streaming software crashed. So let us quickly go back, go back, go back. Yeah. Okay. So introns are excised by DNA's helicase and RNA and the restriction into nucleus, and the answer is the small nuclear RNA. The answer is the small nuclear RNA. I know it got stuck. A very short delay. I'm sorry for that. A very short delay. We'll come back in a second. Okay, moving on to next question, probably the last question. A mutation in the codon which causes a change in the coded amino acid is known as mutation in the codon. It causes a change in the coded amino acid. This is known as mitogenesis, missense mutation, somatic mutation, recombination. And if you check your notes, you will find change in one coded amino acid is the missense mutation. Change in one coded amino acid is the missense mutation, which is the correct answer here. Correct answer is the missense mutation. Okay, moving on. HBS. We had mentioned HBS. HBS has defect in which amino acid position? Your options are 4 of alpha chain, 6 of beta chain, 11 of beta chain, 12 of alpha chain. And the correct answer is 6 of beta chain in the A helix. The glutamic acid is replaced by valine. This is what we had mentioned in HBS, in beta chain, in A helix, position 6. Glutamic acid is replaced by valine. Very good, very good. Previous question, all of you have given the correct answer. It's nice to see. Now, which of the following is not a feature of the genetic code? I told you this is the question where the examiner will very frequently try to stump you, and very frequently they will try to stump you. And your options are degenerate, ambiguous, non-overlapping, non-punctuated. And the answer is ambiguous. It should be unambiguous. The genetic code is unambiguous. It is degenerate, it is non-overlapping, it is non-punctuated. I think I have been able to convey the information about molecular biology very well to all of you. Most of the time, you are giving the correct answer. It is so nice to see all of you participating and giving in the correct answer. Thank you very much for your participation and your active attention. Coming to the next question, the enzymes involved in formation of RNA-induced silencing complex include all of the following except drosha, pasha, dicer, ribozyme. If you recall your notes, the dicer is the other name for DGCR8. When this question was asked for the first time, a lot of the faculty also did not know that Pasha is the other name for DGCR8, and they advised the students that okay, the answer should have been the Pasha. But later on, I told them okay, Pasha is the other name for DGCR8, so that is true. The correct answer is ribozyme. Ribozyme is not involved in the formation of RISC complex. We have the Droshan Pasha, which will remove the cap and the tail, and the dicer, which removes the loop. At most, we have the exportin 5 and the RNA polymerase 2. The ribozyme is nowhere in the picture. Very good. It is so nice to see that all of you have given the correct answer. The correct answer is ribozyme. Correct answer is ribozyme. So that is all for today from my side. Tomorrow we are going to talk about the DNA synthesis, the RNA synthesis, the protein synthesis, and a little bit about regulation. We'll focus at over the lac-operon. We'll see what are the important points that you should know about the lac-operon. We'll talk a little bit about the various techniques. We'll talk about a little bit about the PCR, the RT-PCR, which is being used currently for the COVID infection. Brief overview about you can say. Eliza, if your time permits, and I'll talk a little bit about iron metabolism. The time, I'll tell you what is the time. We'll tell you in the morning. You will be updated about the time for the class. So I'll wait for 2 minutes. If you have any questions, if you want me to repeat any part, then I'll take those questions. Else, we will close for today. If you have any doubts, any queries, any part you want me to repeat, please feel free to send them across.